Okay, I'm going to give you a little tour of this photosynthesis system. This is a system manufactured by Li-6400. They're a company in Lincoln, Nebraska, and they make all sorts of instrumentation for both environmental and genetics research, but they make some of the state-of-the-art gas exchange systems. This instrument was state-of-the-art in the late 90s. This instrument is probably about 20 years old now, but it's still running strong. They're quality instruments. This is the Li-6400. Li-64's actually released a more recent version of this system that's similar called the 6800. But my old Li-64 6400s are still going strong. So, like all gas exchange systems, there is an airtight chamber into which you place the leaf. So when you squeeze the handle, the chamber opens and you place a leaf in there. And I'll show you a little bit more about that in a minute. The infrared gas analyzers are very close to the chamber. They're housed in this part right here, actually behind here. That enables the gases in the chamber to be sensed very rapidly because there's not much distance that the gases have to go before they're analyzed. You can see there's tubing that connects to this main box here. There's pumps in there, and the air enters over here. There's an air entry point right here. Air is sucked in and then run through conditioning chemicals which can scrub CO2 and water vapor out of that airstream. And then there's a part of the instrument here into which you can place a compressed gas cylinder containing CO2. So we can put that cylinder in there, and the instrument, basically what it does is takes air from outside, scrubs all the CO2 out, and then uses the compressed CO2 to add CO2 back in at whatever rate is necessary to maintain your set CO2 concentration in the chamber. This is also the brains of the system, is located in this box. So there's data entry capability, there's a computer in here that logs everything, and then a screen that gives you a readout of important variables. You can control several environmental factors in the chamber, including, I already mentioned, CO2, but there also are steady state units on the side that can allow you to heat or cool the chamber about 5 degrees Celsius above or below ambient. You can also adjust the amount of air that gets bypassed through the dryer column to adjust the humidity in the chamber. Another factor that you can control is light. At the top of the chamber, we'll open it up again, there are a number of LEDs. Now it's a little bit difficult to see it in the video, but those LEDs are red and blue. So if you recall, the primary wavelengths of photosynthesis, or that are absorbed by chlorophyll, are red and blue. So those are the primary wavelengths of the light in that chamber. And you can adjust the intensity of light in that chamber using the controls on the system. Right now, light is very bright. You can see it's very bright. It's basically set to 2,000 micromoles per meter squared per second. That's equivalent to full sunlight. So we can place a leaf into that chamber and then monitor on the system the photosynthesis rate of that leaf, along with a number of other factors, including the stomatal conductance, which is a measure of how wide open the stomata are, the vapor pressure deficit of air in the chamber, and a number of other factors that I'll describe in the lab. So This system is a great tool for measuring photosynthesis rate as well as water relations parameters, because in addition to carbon dioxide infrared gas analyzers, these gas analyzers can also measure water vapor concentration, which allows us to measure transpiration rate of leaves as well as the stomatal conductance of leaves, the degree to which those stomata are open or closed. So in this lab, we're going to use this instrument to measure net photosynthesis of a gymnosperm and an angiosperm. And you'll be able to see how the instrument is used to measure photosynthesis rates.